Protein coenzymes play a role in electron transfer simply because they contain an iron atom, and this iron is present either as iron sulfur cluster or in a heme group. Iron, as you know, can undergo oxidation or reduction. It could be present in the ferric state, which is the oxidized state, plus. It receives an electron to be reduced to the ferrous state, Fe2 plus. The first group of these proteins are called iron sulfur proteins because they contain iron sulfur clusters. And this iron sulfur cluster is made up of iron atoms that are complexed with an equal number of sulfide ions. The two common types you see in the slide are 2Fe2S and 4Fe4S. And no matter what is the type of the cluster, they are always attached to 4 cysteine residues of the protein. The second type of proteins involved in electron transfer are the cytochromes. And the cytochromes are heme-containing proteins. So the proteins, cytochromes, they contain a prosthetic group, what we call as the heme. I'll discuss the structure of the heme in the next slide. And that heme contains an iron atom. That's why these cytochromes can play a role in the electron transfer. As you see, the heme here is made up of a tetraviral structure, 4 viral rings. And the nitrogen atoms of the viral rings, they form coordination bonds with an iron atom. Now, in addition, there are several side chains that are substituents of the viral rings. So that is the general structure of the heme, which is a porphyrin structure. It is a tetraviral structure. Cytochromes are classified into different types, like cytochrome A, cytochrome B, cytochrome C. And these cytochromes, they differ in their absorption spectra because these are cellular pigments. The cytochromes are cellular pigments, so they can absorb the light energy, and they have light absorbing properties, and they differ, as I said, one from the other in the absorption spectra of the heme groups, as well as in the structure of the heme group and the structure of the protein. As far as the structure of the heme, as you see, if you look, for instance, at the cytochrome A, you will find that attached to carbon 2, there is a 17 carbon atom side chain, while in the case of cytochrome B, you will have a vinyl side chain attached to carbon 2. Another difference is shown at carbon 8, while you find in cytochrome A, there is an aldehyde group. You find in cytochrome B, there is a methyl group. Cytochrome B, by the way, has the heme, which is found in the hemoglobin, which is found in myoglobin, as well as in the catalase enzyme. Another difference is how these porphyrin rings are attached to the proteins, because the protein, they could be attached non-covalently or they could be attached covalently, as you see in the case of the cytochrome C heme group. All types of cytochromes are classified on the basis of distinctive light absorbing properties, with those of similar properties segregated into groups designated by lowercase letters A, B, C, and so on. The heme groups of the reduced cytochromes, and those that contain ferrous iron, have a prominent visible absorption spectra consisting of 3 peaks, as you see in the figure, alpha, beta, and gamma. The wavelength of the alpha peak, which varies characteristically with the particular reduced cytochrome species, is useful in differentiating the various cytochromes. Accordingly, the spectra of mitochondrial membranes indicate that they contain 3 cytochrome species, A, B, and C. Within each group of cytochromes, different heme groups environments may be characterized by slightly different alpha peak wavelength. For example, as you will see later, in complex 3, there are 2 type cytochromes. one that absorbs maximally at 5602 nanometers, it's referred to as 562 or BH, that's the high potential, and another one that absorbs maximally at the wavelength 566 and is referred to as BL or B566. As I said earlier, in the inner mitochondrial membrane, there are protein complexes that are associated with the electron transport chain and the production of ATP. And there are four complexes, complex 1, 2, 3, and 4, that include several cofactors, that include several electron carriers that are involved in electron transport, and there is one complex, complex P, that is involved in the synthesis of ATP. This is an overview of these protein complexes forming the electron transport chain. There are four of them, complex 1, 2, 3, and 4. I will go over the systematic names of these complexes as I come to discuss them one by one. And as you see in each complex, there are several electron carriers such as FMN, FAD, iron sulfur clusters, hemes of the cytochromes, or copper ions. I'll be discussing these complexes one by one as I go through. This also shows these complexes again with the cofactors involved in them. Complex 1, for example, as you see in the top of the slide, contains cofactors like NADH and iron sulfur clusters. Complex 2 contains FAD iron sulfur clusters. Complex 3 contains iron sulfur clusters, cytochrome B and cytochrome C1. Complex 4 contains cytochrome A, cytochrome A3, as well as oxygen, which is the terminal electron acceptor. Now, what happens in these complexes? These complexes receive the electrons from either NADH or succinate, as you see here. Let us start with NADH. Complex 1 receives the electrons from NADH. These electrons will pass through a series of carriers that are present in this complex, and then they will eventually reach Q, which will be reduced to QH2, and it will deliver the electrons to the next complex, which is complex 3. Now, complex 3 again has a series of electron carriers that will carry the electrons from QH2, send them to complex 4 via cytochrome C. In other words, cytochrome C shuttles the electrons between complex 3 and complex 4. Now, complex 4 has several electron carriers, and the electrons from there will go to reduce oxygen to water. Oxygen is the terminal acceptor of these electrons. If the electrons come from succinate, when succinate is oxidized to fumarate, that's one of the steps that makes Krebs cycle, as you learned earlier. Then these will pass through complex 2. In complex 2, again, there are several electron carriers that will pass the electrons to complex 3 via quinone. In other words, the quinone will shuttle the electrons between complex 2 and complex 3. And then from cytochrome C, they will go to complex 4 via cytochrome C, and eventually they will reach the oxygen. So the pathway of electrons will be from NADH, complex 1, complex 3, complex 4, eventually to reach oxygen to reduce it to water, or from complex 2 to complex 3 to complex 4, eventually again to reduce oxygen to water. As the electrons pass in the carriers, as you see, they lose some of their energy, and this energy will be used in the creation of a proton gradient. When this proton gradient is dissipated, it will result in the production of ATP. I'll be discussing these details as I go through. This table, as in the previous one, shows the complexes with the different electron carriers that are present in these complexes and which are involved in oxidation reduction reactions. So in complex 1, you see there is iron sulfur clusters. In complex 2, there is iron sulfur clusters. In complex 3, there is cytochrome Bl, cytochrome Bh, and iron sulfur clusters, as well as cytochrome C1. In complex 4, there is cytochrome A, copper atom, A, and there is cytochrome A3, and there is copper B or COB. I'll be discussing all these complexes, as I said, one by one in some detail.